The EFFPAC trial is a randomized controlled trial comparing the investigational product luminor as drug-coated balloon with the standard conventional POBA plain old balloon angioplasty. Today we are looking at the 24-month results, and the primary endpoint was actually the late lumen loss, which we already presented last year with astonishing results, a very low rate of late lumen loss for the investigational product with 0.14 as compared to 1 millimeter in the POBA group. And today we are looking at the secondary endpoints. That means patency rate, freedom from TLR, binary restenosis, and also the other clinical parameters like change of Rutherford, change of ABI as compared to the control group. We were able to maintain the great results from the 12 months to the 24-month results. So the freedom from TLR rate is still with 97%, the highest which was so far reached in clinical trials for this same area. And also the patency rate with 90%, which is also very high. Binary restenosis with 20% versus 46 or 47% in the POBA group, also astonishing results. So the efficacy is very, very high of this investigational product of the luminor balloon, and also the clinical measures, so quality of life of the patient, it was no more significant, but still there's a strong tendency towards better quality of life of the patient. And this is something which is quite important today. Well, if you would have asked me 1 year ago, that was not so much important to look at all cause of mortality, because at 24-month, actually other trials showed a tendency towards all cause of death, but in our trial we actually could show that there is no real increased risk with our investigational product. So that means in numbers we actually had 1 death in the DCB group, non-related death in the DCB group, and 2 non-related death in the control group, POBA group, and therefore our risk ratio is below 1. We could prove that well, to look at all cause of death, which is actually none of the trials, this is also true for EFFPAC, is powered for all cause of death. But still, nowadays after the Casanos meta-analysis, everyone talks about it, because most of the trials, not all, but most of the trials showed a certain tendency towards all cause of death. But actually in our trial it was not, we could not confirm this. We actually only had 1 case of death in the non-related death in the DCB group as compared to 2 deaths in the POBA group, meaning a risk ratio below 1. Well, statistically seen, there is no difference between both groups. That means also that there is no risk. What makes the luminor different? This is actually not only the case that we're using paclitaxel, all the others are also using paclitaxel, but also the excipient. But mainly, I believe, I do not have scientific proof for it, but from our data, I can only say that it is very effective, and I think that the effectiveness has to do with the coating technology. I think the uptake in the vessel wall should be somehow higher than as compared to other balloons, and the loss for transfer to the target lesion is probably less. So that is, but this is only estimation, because we do not have scientific proof for it. But actually, our clinical results show that the balloon must, for some reason, be more effective than others, and I believe that it's due to the nanotechnology used in this balloon.